Hello, my name is Linda, and I will be presenting about gene therapy, more specifically exploring the effectiveness of gene therapy and personalized medicine for autism spectrum disorder. This is a topic that really interests me, as it is also a major focus in current research regarding the interventions that can be carried out for the treatment and identification of ASD. So I'll be exploring that. So the content of this presentation will have an introduction to ASD and then an overview of genetic therapy and its current state, as well as the implication of personalized medicine and gene therapy specifically. So I'll be focusing on those two major core parts in this presentation. So let's start with an introduction to ASD. So autism spectrum disorder, as we can see, is a very prevalent, significant um, it's a very complex neurodevelopmental disorder that affects a significant portion of the population. About 1.5% of the global population is affected by ASD, which is actually a very big number, and therefore um, it is a very complex NDD, which is caused by neurological, genetic, primarily genetic, and environmental factors. Going to the current interventions of ASD. So currently we have lots of different current interventions, but I've picked out, for example, applied behavior analysis, which is a psychological intervention that seeks to changing the behavior of social significance, which shows that the intervention is more focused on environmental factors than genetic factors. We see some of the environmental factors leading to ASD. However, it's not able to address some of the genetic factor. The other is antipsychotics. So antipsychotics is a class of psychotic medication that is being taken that is used to manage psychosis and psychotic disorders. So um, we see that there are lots of side effects for this intervention, like it could be weight loss that may lead to diabetes. There's also blurred vision, as well as dry mouth and dizziness. We see a lot. We see that this. So we see that current interventions, um, they face a challenge in that the individual at an individual level, ASD is very different from person to person, and it varies even at the same level for individuals. And this is because many genetic and environmental factors come into play for including the gene expression which makes it very vary for different people. Therefore, it is very important to have a more sophisticated, individualized approach for its identification and its treatment, which is a very important um, intervention point that we need to take based on the genetic profiles of the individuals. Therefore, we need a paradigm shift in the intervention, and that's how gene therapy comes into play. So we see that there are lots of genetic factors, and they're mainly the ones that drive ASD development. So gene therapy is a genetic intervention that treats and identifies genetic factors that leads to ASD. So they are this the this genetic intervention involves editing the gene or genome editing, and that involves deleting, or it could be inserting and altering the gene expression of the gene so that it can be through the process of gene replacement or through gene silencing that we can identify modify the gene expression. So by using this technique, we kind of can tackle the core problem, the major cause for ASD. Therefore, this becomes very apparent in monogenic ASDs because they're caused by a single mutated gene. Therefore, for its intervention, it will be targeted on that specific gene, and therefore, in order to reverse the effects. However, for more complex ones, for example, polygenic ASDs that are caused by multiple genes that are responsible for the ASD that have been mutated, it would be harder to target those genes, and it may also lead to off-target effects as well. And therefore, for polygenic ASDs, it would be harder, and therefore more advanced technologies would be needed in order to better treat the ASD. Therefore, we have seen that preclinical studies with monogenic ASDs have been progressing quite effectively, especially with gene delivery techniques to the central nervous system related to gene silencing and replacement, in general, genome editing in the central nervous system. And therefore, we see that having advancements in the monogenic ASD would enable us, in a way, to advance to polygenic ASDs that are more complex and hard to intervene on. So we see that gene therapy can be carried out at different levels. There's the DNA level, mRNA level, and protein level. We tend to focus more on the DNA level. So at the DNA level, we have the CRISPR technology that is being used and has been there has been a huge amount of progress in this technology, and it's a very modern, advanced technology that researchers have been using in order to edit the gene. So CRISPR involves, so basically this technology has two key elements. So the first is the guide RNA, which aligns with the targeted cell that contains the mutated gene that needs to be edited. And the second is the Cas9, which is the one that does the editing, and therefore it induces DNA that can cut or break the gene sequence. And then we also have in parallel mRNA level and protein level, which are more focused on having precise and targeted intervention. So they more tend to focus on a targeted approach for interventions. So they would directly target the mutated gene, so that it would have a less effective off-target on the ways, as it has a lot of risk for the patient. As editing a gene that has not been targeted or has not mutated can lead to very huge negative consequences. Therefore, these are the three levels that we use that gene therapy that the intervention focuses on. These are the different technologies that we find tend to intervene at these three different levels. The central dogma. So we see that these advancements, they highlight the dynamic nature of gene therapy, as it is a very promising approach to address very complex genetic factors. But at the same time, they all have their own challenges that they face. And therefore, we see that some of the three major challenges that I've highlighted are, firstly, we have the off-target effect, which is something I already mentioned. That is the unintended point mutation, or deletion or insertion or relocation of genes, and therefore has very bad consequences. As the Cas9 enzyme would target a gene that has not mutated, or a normal gene, which would, instead of, that's why we technically need targeted interventions that directly and effectively intervene on the targeted mutated gene. And the second is the means of delivery, um, because it is very hard to physically transport the Cas enzyme to the central nervous system, because in the case of ASD, the genetic interventions are on the brain cells, and the brain cells are very sensitive, and it needs cautious intervention for its treatment or its identification. And therefore there are lots of different means of delivery, like viral and non-viral vectors that may or may not involve viruses to transport the Cas9 enzyme to the targeted gene. And therefore, coupled with that, we also have the possibility of having an off-target effect, which also makes it even harder. Therefore, we have to consider to make it cautious to lessen the risk as a means of delivery, but at the same time target the specific gene that has been mutated. And therefore, this shows the big challenge that we face, as both challenges are interlinked and they're really very hard to solve very easily, and therefore there is a need for more advanced technologies that can tackle these challenges. And also we have ethical concerns on genetic interventions, as we see that um, having people with with autism spectrum disorder being treated by inserting genes, that itself raises a lot of ethical issues in the community, which is something to be aware of for researchers, um, as it is something that really needs to be considered in the interventions and in how it is being conveyed for the community. Um, Okay, so with that being said, we also have another type of intervention, that is environmental intervention. It's quite different from genetic intervention because it focuses on intervening on the environmental factors that lead to ASD. So they tend to more focus on enhancing social skills, their effectiveness and broader applicability, and especially how to live with society and um, how to interact with the environment, and therefore is basically tackling the environmental factors related to ASD. And consider that there are lots of different effective interventions that have been carried out. However, it does not fully address the wide range of symptoms and functional impairments, as we said, that is laid out in a spectral form. And the major reason for it is the genetic factors. Therefore, it does not address the core reason or the core cause for ASD, and therefore, even though it's effective, it does not fully address that disorder. However, we see that the environmental and genetic factors actually overlap. We see that environmental factors actually affect negative genetic factors, which is the reason why people have lots of different, um, which is the reason why people have different symptoms and react very differently. Um, and this is mainly because the gene expression is also affected by the background of the individual and the environment that the individual has been in, and therefore there are lots of different factors that we need to consider when thinking about the different factors, and therefore we will have different individual profiles, genetic profiles for different individuals. Um, Therefore, this necessitates a more personalized approach that we can take for gene therapy. That's how precision medicine comes into play. In other words, it's also called personalized medicine, and therefore it considers the individual factors and the variety of symptoms that people can show regarding ASD. So therefore, um, it focuses on a more holistic approach that we can take, and having individual genetic profile. Therefore, by customizing the treatments based on the different individual profiles and the different genetic makeups of the individual, um, personalized medicine or precision medicine will have the potential to actually revolutionize gene therapy to be based on the patient's profile. And therefore, one of the major focus for the clinical trials was identifying biomarkers that will be able to categorize individuals based on their genetic profile. So those having kind of this similar genetic profile will be categorized as one, and therefore it makes the interventions more effective and easily made as well. Therefore, this can encompass lots of different things and lots of different features of the human imaging as well, and therefore the genetic profile constitutes different aspects of the factors for ASD. All right, so in conclusion, wrapping all the presentation, we see that gene therapy has a lot of potential in the treatment identification of ASD. So throughout the presentation, I've seen lots of different aspects of gene therapy and also how it contrasts with current interventions and environmental interventions as well. So we can see that generally we can say that gene therapy has a lot of potential, and we'll see how the research goes around it and how we'll be able to revolutionize the treatment for ASD. Thank you for listening.